today I'll be talking about what happens if we would eat this meal every single day continuously. So our arteries are like pipes. If we don't maintain them well, they tend to get clogged over time. This gradual clogging of the artery is called atherosclerosis. So over time, as you can see here, this is a picture of an artery, and over time there is something called plaque, which is this yellow gooey material here that accumulates, and it's made of fat, cholesterol, debris, and this can thicken and rupture, leading to different heart outcomes like heart attack or stroke. Atherosclerosis underlies several cardiovascular diseases, and cardiovascular diseases is one of the leading causes of death and disability worldwide. And this is the 100th year that cardiovascular diseases have been the leading cause of death. And within the next 10 minutes, by the end of my talk, there will be 15 people in US alone who would have died because of heart diseases. So this is a really, really scary number. So what puts us at risk for heart diseases? Well, there are several risk factors, some of which we cannot change, like genetics, but other things like healthy eating and exercising can even prevent heart diseases. The problem is that most of the clinical markers are focusing on the advanced stage. Once the plaque has already built up, there is already a constriction or obstruction in the blood flow, and by then it might be a little too late. Whereas there's a lack of a reliable marker in the early stage before any symptoms develop or any symptoms manifest. So, and roughly 25% of the cardiovascular disease patients do not even display any of the classical clinical markers either. So there is a real need for understanding more. So our first question was, can we categorize people based on their risk for developing atherosclerosis or the disease progression before any of these symptoms manifest? I know the talk has been a little downer so far, but yes, there is promise. We can actually categorize people. So how do we do this? We actually employ a high-fat meal challenge. So we administer a typical Western-style high-fat meal to subjects from undergraduate students who are really healthy, biking every day, to people who are moderately obese. But none of these people are particularly taking medications for their diseases. So these are mostly healthy people. And then we collect blood. So we collect blood 3.5 hours after we administer the meal, and then isolate something called the dietary lipoproteins. Dietary lipoproteins are basically lipid-rich particles. These particles package fatty acids that we eat and circulate it to different organs which need energy, like muscle, or that store energy, like fat tissues. And the particular lipoprotein we are focusing on is this triglyceride-rich lipoprotein, which for the rest of my talk I'll be addressing as TGRL. And these lipoproteins are commonly referred to as bad fat or bad cholesterol. So when you read magazines, they usually call this as bad cholesterol. And what they do is they are very enriched in triglycerides, and they are upregulated after a meal. And they even have a causal relationship for the plaque buildup and are one of the independent risk factor for cardiovascular diseases. So once we isolate these lipoproteins, we add it to our ex vivo model of our blood vessel or our artery model that's outside of the body. So we use this because we can't really go inside our body and study what's happening. So we use endothelial cells that line up this region of the artery wall, which is very interesting because that's the most, that's where there's a lot of interaction with other cells and lipoprotein circulation, etc. And we can also use this to model what would happen if there's excessive circulation of TGRL or other cell interactions here. And this is a figure of how our setup looks. So here we can also induce different flow conditions and then see how are the cells, which would be at the bottom right here, they'll be like that, the cartoon figure, right there, how would they respond in different conditions? So this is our artery model. And then this is an image of how it would look under a microscope, where these are cells and the green droplets or green particles are actually the lipoproteins that are taken up by the endothelial cells. And you can see, so it is a model of what would happen inside of our body. And then we look at some of the interesting functional outcomes, which I'll talk about next. So the particular marker we are very interested in is VCAM1 expression. VCAM is adhesion molecule, and it is highly expressed in plaque areas. And what it does is, the more endothelial cells express VCAM1, I would think of it as the more sticky the cell gets. So it attaches to, or immune cells or adhere to the endothelial cells over time. And this is an example of how it looks in our model. So this would be, again, endothelial cells, and all the white circular cells are immune cells that adhere to the endothelial cells that actually is the artery wall lining. And why this is important is because this interaction is one of the early steps or early events for atherosclerosis. Over time, the more immune cells adhere and transmigrate, it kind of accumulates into the plaque area as well. So it's a very important functional marker that we are looking at. And then coming to the interesting part, we can classify people based on how they respond. So not everyone eats the same meal and responds the same way. And this isn't that surprising, but what is really cool is that now, after lipoprotein is added to the endothelial cells, we can see that the lipoprotein from some subjects, some humans, increases this VCAM1 marker, and that increases these immune cells adhesion in the endothelial cells. Whereas some other people whose lipoprotein decreases VCAM1 expression and reduces the number of immune cells that are adhered on the endothelial surface. So this is really interesting because now you have like a individualized, personalized tool that can categorize people based on their risk for disease progression. So our next question was, if everyone had the exact same meal, why are the responses so different? We were thinking it's probably what's inside these particles itself. So the lipoprotein composition is very much reflective of a person's metabolism and also what they have been eating on a regular basis. So not just the meal, but the diet they have been consuming as well. And so for this, we measured about 153 fatty acid metabolites in the particles itself. And then we used that to see if the subject or the lipoproteins clustered together based on their compositional difference, and it does. And further from here, so this helped us narrow to about a subset of 25 different fatty acid metabolites that were key in differentiating the composition between these two lipoprotein groups. And we went ahead and made a mathematical model that would help us predict which category these people would fall in by just looking at the composition of these particles. So we can think of this as, let's say there's a blood test where we can collect a person's blood, isolate the lipoproteins, look at the subset of fatty acid metabolites, and then see if we can categorize them into a pro-responder category or an anti-responder category based on just their composition. So the third thing is, one thing that would be really cool is, can I use this as a marker for my daily lifestyle changes? So can I, obviously there isn't one diet fits all, there isn't one exercise fits all. Can we use this to maybe find the best regimen possible for that individual person? And so that was what we were looking at next. So this is a representative data from subject number 2. It is the same person has been given 2 separate diets, and then we looked at how our marker changes over time. So this is the before the meal time point. So they're fasting. This, it's the same person. He's starting around the same time point or same baseline. But after the meal, after a high-fat meal, you can see it increase, whereas after the low-fat meal, you can see this decrease. So there is, so every small changes can be tracked with the help of this marker. So in conclusion, I want to stress that we have identified a marker that can categorize people based on their risk factor for disease progression. Then we have built a computational model or a mathematical model where we can use the composition of these particles to then predict which categories they might fall into. And hopefully with additional validation, recruiting people who might be taking medications, we could use this in the clinics for for a healthy lifestyle, preventative care, or even for putting people on different therapeutic regimens. Next, I would like to thank my lab members who have always been helpful. They listen to me talk about fatty acids all the time. I would particularly like to thank Dr. Pasrini, who is my advisor, and all the undergraduates who helped me a lot with my project, and also the funding sources. Thank you.